Biogeography. Biogeography is the study of the distribution of species and ecosystems in geographic space and through geological time. Organisms and biological communities often vary in a regular fashion along geographic gradients of latitude, elevation, isolation and habitat area. 1. Phytogeography is the branch of biogeography that studies the distribution of plants. Zoogeography is the branch that studies distribution of animals. Mycogeography is the branch that studies distribution of fungi, such as mushrooms. Frontispiece to Alfred Russel Wallace's book The Geographical Distribution of Animals. Knowledge of spatial variation in the numbers and types of organisms is as vital to us today as it was to our early human ancestors, as we adapt to heterogeneous but geographically predictable environments. Biogeography is an integrative field of inquiry that unites concepts and information from ecology, evolutionary biology, taxonomy, geology, physical geography, paleontology, and climatology. 2, 3. Modern biogeographic research combines information and ideas from many fields, from the physiological and ecological constraints on organismal dispersal to geological and climatological phenomena operating at global spatial scales and evolutionary timeframes. The short-term interactions within a habitat and species of organisms describe the ecological application of biogeography. Historical biogeography describes the long-term, evolutionary periods of time for broader classifications of organisms. 4 early scientists, beginning with Carl Linnaeus, contributed to the development of biogeography as a science. The scientific theory of biogeography grows out of the work of Alexander von Humboldt, 1769-1859-5 Francisco José de Caldas, 1768-1816, 6 Hewitt Cottrell Watson, 1804-1881, 7 Alphonse de Candolle, 1806-1893, 8 Alfred Russel Wallace, 1823-1913, 9 Philip Lutley Sclater, 1829-1913, and other biologists and explorers. 10. Introduction. The patterns of species distribution across geographical areas can usually be explained through a combination of historical factors such as speciation, extinction, continental drift, and glaciation. Through observing the geographic distribution of species, we can see associated variations in sea level, river routes, habitat, and river capture. Additionally, this science considers the geographic constraints of landmass areas and isolation, as well as the available ecosystem energy supplies. Over periods of ecological changes, biogeography includes the study of plant and animal species in their past and/or present living refugium habitat, their interim living sites, and/or their survival locales. 11. As writer David Quammen put it, biogeography does more than ask which species and where. It also asks why, and, what is sometimes more crucial, why not? 12. Modern biogeography often employs the use of geographic information systems, GIS, to understand the factors affecting organism distribution, and to predict future trends in organism distribution. 13. Often mathematical models and GIS are employed to solve ecological problems that have a spatial aspect to them. 14. Biogeography is most keenly observed on the world's islands. These habitats are often much more manageable areas of study because they are more condensed than larger ecosystems on the mainland. 15. Islands are also ideal locations because they allow scientists to look at habitats that new invasive species have only recently colonized and can observe how they disperse throughout the island and change it. They can then apply their understanding to similar but more complex mainland habitats. Islands are very diverse in their biomes, ranging from the tropical to Arctic climates. This diversity in habitat allows for a wide range of species study in different parts of the world. One scientist who recognized the importance of these geographic locations was Charles Darwin, who remarked in his journal, "The zoology of archipelagos will be well worth examination." 15. 2 chapters in On the Origin of Species were devoted to geographical distribution. History. 18th century. The first discoveries that contributed to the development of biogeography as a science began in the mid-18th century, as Europeans explored the world and described the biodiversity of life. During the 18th century, most views on the world were shaped around religion and for many natural theologists, the Bible. Carl Linnaeus, in the mid-18th century, improved our classifications of organisms through the exploration of undiscovered territories by his students and disciples. When he noticed that species were not as perpetual as he believed, he developed the mountain explanation to explain the distribution of biodiversity. When Noah's Ark landed on Mount Ararat and the waters receded, the animals dispersed throughout different elevations on the mountain. This showed different species in different climates, proving species were not constant. 4. Linnaeus' findings set a basis for ecological biogeography. Through his strong beliefs in Christianity, he was inspired to classify the living world, which then gave way to additional accounts of secular views on geographical distribution. 10. He argued that the structure of an animal was very closely related to its physical surroundings. This was important to Georges-Louis Buffon's rival theory of distribution. 10. Closely after Linnaeus, Georges-Louis Leclerc, Comte de Buffon observed shifts in climate and how species spread across the globe as a result. He was the first to see different groups of organisms in different regions of the world. Buffon saw similarities between some regions which led him to believe that at one point continents were connected and then water separated them and caused differences in species. His hypotheses were described in his work, the 36-volume Histoire naturelle, générale et particulière, in which he argued that varying geographical regions would have different forms of life. This was inspired by his observations comparing the Old and New World, as he determined distinct variations of species from the two regions. Buffon believed there was a single species creation event, and that different regions of the world were homes for varying species, which is an alternate view than that of Linnaeus. Buffon's law eventually became a principle of biogeography by explaining how similar environments were habitats for comparable types of organisms. 10. Buffon also studied fossils, which led him to believe that the Earth was over tens of thousands of years old, and that humans had not lived there long in comparison to the age of the Earth. 4. 19th century. Following the period of exploration came the Age of Enlightenment in Europe, which attempted to explain the patterns of biodiversity observed by Buffon and Linnaeus. At the birth of the 19th century, Alexander von Humboldt, known as the founder of plant geography, developed the concept of physique générale to demonstrate the unity of science and how species fit together. As one of the first to contribute empirical data to the science of biogeography through his travel as an explorer, he observed differences in climate and vegetation. The Earth was divided into regions which he defined as tropical, temperate, and Arctic, and within these regions there were similar forms of vegetation. 4. This ultimately enabled him to create the isotherm, which allowed scientists to see patterns of life within different climates. 4. He contributed his observations to findings of botanical geography by previous scientists, and sketched this description of both the biotic and abiotic features of the Earth in his book, Cosmos. 10. Augustin de Candolle contributed to the field of biogeography as he observed species competition and the several differences that influenced the discovery of the diversity of life. He was a Swiss botanist and created the first laws of botanical nomenclature in his work, Prodromus. 16. He discussed plant distribution, and his theories eventually had a great impact on Charles Darwin, who was inspired to consider species adaptations and evolution after learning about botanical geography. De Candolle was the first to describe the differences between the small-scale and large-scale distribution patterns of organisms around the globe. 10. Several additional scientists contributed new theories to further develop the concept of biogeography. Charles Lyell developed the theory of uniformitarianism after studying fossils. This theory explained how the world was not created by one sole catastrophic event, but instead from numerous creation events and locations. 17. Uniformitarianism also introduced the idea that the Earth was actually significantly older than was previously accepted. Using this knowledge, Lyell concluded that it was possible for species to go extinct. 18. Since he noted that Earth's climate changes, he realized that species distribution must also change accordingly. Lyell argued that climate changes complemented vegetation changes, thus connecting the environmental surroundings to varying species. This largely influenced Charles Darwin in his development of the theory of evolution. 10. Charles Darwin was a natural theologist who studied around the world, and most importantly in the Galapagos Islands. Darwin introduced the idea of natural selection, as he theorized against previously accepted ideas that species were static or unchanging. His contributions to biogeography and the theory of evolution were different from those of other explorers of his time, because he developed a mechanism to describe the ways that species changed. His influential ideas include the development of theories regarding the struggle for existence and natural selection. Darwin's theories started a biological segment to biogeography and empirical studies, which enabled future scientists to develop ideas about the geographical distribution of organisms around the globe. 10. Alfred Russel Wallace studied the distribution of flora and fauna in the Amazon basin and the Malay archipelago in the mid-19th century. His research was essential to the further development of biogeography, and he was later nicknamed the father of biogeography. Wallace conducted fieldwork researching the habits, breeding and migration tendencies, and feeding behavior of thousands of species. He studied butterfly and bird distributions in comparison to the presence or absence of geographical barriers. His observations led him to conclude that the number of organisms present in a community was dependent on the amount of food resources in the particular habitat. 10. Wallace believed species were dynamic by responding to biotic and abiotic factors. He and Philip Sclater saw biogeography as a source of support for the theory of evolution as they used Darwin's conclusion to explain how biogeography was similar to a record of species inheritance. 10. Key findings, such as the sharp difference in fauna either side of the Wallace line, and the sharp difference that existed between North and South America prior to the relatively recent faunal interchange, can only be understood in this light. Otherwise, the field of biogeography would be seen as a purely descriptive one. 4. 20th and 21st century. Schematic distribution of fossils on Pangaea according to Wegener. Moving on to the 20th century, Alfred Wegener introduced the theory of continental drift in 1912, though it was not widely accepted until the 1960s. 4. This theory was revolutionary because it changed the way that everyone thought about species and their distribution around the globe. The theory explained how continents were formerly joined in one large landmass, Pangaea, and slowly drifted apart due to the movement of the plates below Earth's surface. The evidence for this theory is in the geological similarities between varying locations around the globe, the geographic distribution of some fossils including the mesosaurs on various continents, and the jigsaw puzzle shape of the land masses on Earth. Though Wegener did not know the mechanism of this concept of continental drift, this contribution to the study of biogeography was significant in the way that it shed light on the importance of environmental and geographic similarities or differences as a result of climate and other pressures on the planet. Importantly, late in his career Wegener recognized that testing his theory required measurement of continental movement rather than inference from fossil species distributions. 19. In 1958 paleontologist Paul S. Martin published a biogeography of reptiles and amphibians in the Gomez Farias region, Tamaulipas, Mexico, which has been described as groundbreaking, 2035 p. and a classic treatise in historical biogeography. 21 311 p. Martin applied several disciplines including ecology, botany, climatology, geology, and Pleistocene dispersal routes to examine the herpetofauna of a relatively small and largely undisturbed area, but ecologically complex, situated on the threshold of temperate, tropical, Nearctic and neotropical regions, including semi-arid lowlands at 70 meters elevation and the northernmost cloud forest in the western hemisphere at over 2,200 meters. 20, 21, 22. Biologist Edward O. Wilson, CO authored the theory of island biogeography, which helped in stimulating much research on this topic in the late 20th and 21st centuries. The publication of the theory of island biogeography by Robert MacArthur and E. O. Wilson in 1967-23 showed that the species richness of an area could be predicted in terms of such factors as habitat area, immigration rate and extinction rate. This added to the long-standing interest in island biogeography. The application of island biogeography theory to habitat fragments spurred the development of the fields of conservation biology and landscape ecology. 24. Classic biogeography has been expanded by the development of molecular systematics, creating a new discipline known as phylogeography. This development allowed scientists to test theories about the origin and dispersal of populations, such as island endemics. For example, while classic biogeographers were able to speculate about the origins of species in the Hawaiian Islands, phylogeography allows them to test theories of relatedness between these populations and putative source populations on various continents, notably in Asia and North America. 15. Biogeography continues as a point of study for many life sciences and geography students worldwide, however it may be under different broader titles within institutions such as ecology or evolutionary biology. Continues as a point of study for many life sciences and geography students worldwide, however it may be under different broader titles within institutions such as ecology or evolutionary biology. In recent years, one of the most important and consequential developments in biogeography has been to show how multiple organisms, including mammals like monkeys and reptiles like squamates, overcame barriers such as large oceans that many biogeographers formerly believed were impossible to cross. 25. See also oceanic dispersal. Modern applications. Biogeographic regions of Europe. Biogeography now incorporates many different fields including but not limited to physical geography, geology, botany and plant biology, zoology, general biology, and modeling. A biogeographer's main focus is on how the environment and humans affect the distribution of species as well as other manifestations of life such as species or genetic diversity. Biogeography is being applied to biodiversity conservation and planning, projecting global environmental changes on species and biomes, projecting the spread of infectious diseases, invasive species, and for supporting planning for the establishment of crops. Technological evolving and advances have allowed for generating a whole suite of predictor variables for biogeographic analysis, including satellite imaging and processing of the Earth. 26. Two main types of satellite imaging that are important within modern biogeography are Global Production Efficiency Model, GLOBPEM, and Geographic Information Systems, GIS. GLOBPEM uses satellite imaging gives repetitive, spatially contiguous, and time-specific observations of vegetation. These observations are on a global scale. 27. GIS can show certain processes on the Earth's surface like whale locations, sea surface temperatures, and bathymetry. 28. Current scientists also use coral reefs to delve into the history of biogeography through the fossilized reefs. 2. Global information systems are either dedicated to, or have strong focus on, biogeography in the form of the spatial location of observations of organisms, namely the Global Biodiversity Information Facility, GBIF. 2.57 billion species occurrence records reported as of August 2023, 29, and, for marine species only, the Ocean Biodiversity Information System, OBIS, originally the Ocean Biogeographic Information System, 116 million species occurrence records reported as of August 2023. 30. While at a national scale, similar compilations of species occurrence records also exist such as the UK National Biodiversity Network, the Atlas of Living Australia, and many others. In the case of the oceans, in 2017 Costello et al. Analyzed the distribution of 65,000 species of marine animal sand plants as then documented in OBIS, and used the results to distinguish 30 distinct marine realms, split between continental shelf and offshore deep sea areas. 31. Since it is self-evident that compilations of species occurrence records cannot cover with any completeness areas that have received either limited or no sampling, a number of methods have been developed to produce arguably more complete, predictive, or modeled distributions for species based on their associated environmental or other preferences, such as availability of food or other habitat requirements. This approach is known as either environmental niche modeling (ENM) or species distribution modeling (SDM). Depending on the reliability of the source data and the nature of the models employed, including the scales for which data are available, maps generated from such models may then provide better representations of the real biogeographic distributions of either individual species, groups of species, or biodiversity as a whole. However, it should also be borne in mind that historic or recent human activities, such as hunting of great whales, or other human-induced exterminations, may have altered present-day species distributions from their potential, full, ecological footprint. Examples of predictive maps produced by niche modeling methods based on either GBIF terrestrial or OBIS marine, plus some freshwater data are the former LifeMapper project at the University of Kansas, now continued as a part of BiodaFi 32, and AquaMaps, which is a 2023 contained model distributions for around 200,000 terrestrial and 33,000 species of teleosts, marine mammals and invertebrates, respectively. 32-33. One advantage of ENM SDM is that in addition to showing current, or even past model distributions, insertion of changed parameters such as the anticipated effects of climate change can also be used to show potential changes in species distributions that may occur in the future based on such scenarios. 34. Paleobiogeography. Distribution of 4 Permian and Triassic fossil groups used as biogeographic evidence for continental drift and land bridging. Paleobiogeography goes one step further to include paleogeographic data and considerations of plate tectonics. Using molecular analyses and corroborated by fossils, it has been possible to demonstrate that perching birds evolved first in the region of Australia or the adjacent Antarctic, which at that time lay somewhat further north and had a temperate climate. From there, they spread to the other Gondwanan continents and Southeast Asia, the part of Laurasia then closest to their origin of dispersal, in the late Paleogene, before achieving a global distribution. Distribution of 4 Permian and Triassic fossil groups used as biogeographic evidence for continental drift and land bridging. Paleobiogeography goes one step further to include paleogeographic data and considerations of plate tectonics. Using molecular analyses and corroborated by fossils, it has been possible to demonstrate that perching birds evolved first in the region of Australia or the adjacent Antarctic, which at that time lay somewhat further north and had a temperate climate. From there, they spread to the other Gondwanan continents and Southeast Asia, the part of Laurasia then closest to their origin of dispersal, in the late Paleogene, before achieving a global distribution in the early Neogene. 35. Not knowing that at the time of dispersal, the Indian Ocean was much narrower than it is today, and that South America was closer to the Antarctic, one would be hard-pressed to explain the presence of many ancient lineages of 35. Not knowing that at the time of dispersal, the Indian Ocean was much narrower than it is today, and that South America was closer to the Antarctic, one would be hard-pressed to explain the presence of many ancient lineages of perching birds in Africa, as well as the mainly South American distribution of the Sabasans. Paleobiogeography also helps constrain hypotheses on the timing of biogeographic events such as vicariance and geodispersal, and provides unique information on the formation of regional biotas. For example, data from species-level phylogenetic and biogeographic studies tell us that the Amazonian teleost fauna accumulated in increments over a period of tens of millions of years, principally by means of allopatric speciation, and in an arena extending over most of the area of tropical South America. Albert and Rees 2011. In other words, unlike some of the well-known insular faunas, Galapagos finches, Hawaiian drosophilid flies, African rift lake cichlids, the species-rich Amazonian ichthyofauna is not the result of recent adaptive radiations. 36. For freshwater organisms, landscapes are divided naturally into discrete drainage basins by watersheds, episodically isolated and reunited by erosional processes. In regions like the Amazon Basin, or more generally Greater Amazonia, the Amazon Basin, Orinoco Basin, and Guianas with an exceptionally low, flat topographic relief, the many waterways have had a highly reticulated history over geological time. In such a context, stream capture is an important factor affecting the evolution and distribution of freshwater organisms. Stream capture occurs when an upstream portion of one river drainage is diverted to the downstream portion of an adjacent basin. This can happen as a result of tectonic uplift or subsidence, natural damming created by a landslide, or headward or lateral erosion of the watershed between adjacent basins. 36. Concepts and fields. Biogeography is a synthetic science, related to geography, biology, soil science, geology, climatology, ecology and evolution. Some fundamental concepts in biogeography include allopatric speciation, the splitting of a species by evolution of geographically isolated populations. Evolution, change in genetic composition of a population. Extinction, disappearance of a species. Dispersal, movement of populations away from their point of origin, related to migration endemic areas. Geodispersal, the erosion of barriers to biotic dispersal and gene flow, that permit range expansion and the merging of previously isolated biotas. Range and distribution. Vicariance, the formation of barriers to biotic dispersal and gene flow, that tend to subdivide species and biotas, leading to speciation and extinction. Vicariance biogeography is the field that studies these patterns. Comparative biogeography. The study of comparative biogeography can follow 2 main lines of investigation. 37. Systematic biogeography, the study of biotic area relationships, their distribution, and hierarchical classification. Evolutionary biogeography, the proposal of evolutionary mechanisms responsible for organismal distributions. Possible mechanisms include widespread taxa disrupted by continental breakup or individual episodes of long-distance movement. Biogeographic regionalizations. There are many types of biogeographic units used in biogeographic regionalization schemes, 38, 39, 40, as there are many criteria, species composition, physiognomy, ecological aspects, and hierarchization schemes. Biogeographic realms, ecozones, bioregions, sensu stricto, ecoregions, zoogeographical regions, floristic regions, vegetation types, biomes, etc. The terms biogeographic unit, 41, biogeographic area, 42, can be used for these categories, regardless of rank. In 2008, an international code of area nomenclature was proposed for biogeography. 43, 44, 45, it achieved limited success. Some studies commented favorably on it, but others were much more critical. 46, and it has not yet gained a significant following. 47, similarly, a set of rules for paleobiogeography, 48, has achieved limited success. 47, 49, in 2000, Westerman suggested that the difficulties in getting formal nomenclatural rules established in this field might be related to the curious fact that neither Palean or neo-biogeographers are organized in any formal groupings or societies, nationally, so far as I know, or internationally, an exception among active disciplines. 50. See also Allen's rule, Bergmann's rule. Quantitative ecology. Quantitative ecology is the application of advanced mathematical and statistical tools to any number of problems in the field of ecology. It is a small but growing subfield in ecology, reflecting the demand among practicing ecologists to interpret ever larger and more complex data sets using quantitative reasoning. Quantitative ecologists might apply some combination of deterministic or stochastic mathematical models to theoretical questions, or they might use sophisticated methods in applied statistics for experimental design and hypothesis testing. Typical problems in quantitative ecology include estimating the dynamics and status of wild populations, modeling the impacts of anthropogenic or climatic change on ecological communities, and predicting the spread of invasive species or disease outbreaks. Quantitative ecology, which mainly focuses on statistical and computational methods for addressing applied problems, is distinct from theoretical ecology, which tends to focus on understanding the dynamics of simple mechanistic models and their implications for a general set of biological systems using mathematical arguments. See also Taylor's law, theoretical ecology, thermal ecology. Mathematical and theoretical biology. Mathematical and theoretical biology, or biomathematics, is a branch of biology which employs theoretical analysis, mathematical models and abstractions of living organisms to investigate the principles that govern the structure, development and behavior of the systems, as opposed to experimental biology, which deals with the conduction of experiments to test scientific theories. 1. The field is sometimes called mathematical biology or biomathematics to stress the mathematical side, or theoretical biology to stress the biological side. 2. Theoretical biology focuses more on the development of theoretical principles for biology, while mathematical biology focuses on the use of mathematical tools to study biological systems, even though the two terms are sometimes interchanged. 3. 4. Yellow chamomile head showing the Fibonacci numbers in spirals consisting of 21 blue and 13 aqua. Such arrangements have been noticed since the Middle Ages and can be used to make mathematical models of a wide variety of plants. Mathematical biology aims at the mathematical representation and modeling of biological processes, using techniques and tools of applied mathematics. It can be useful in both theoretical and practical research. Describing systems in a quantitative manner means their behavior can be better simulated, and hence properties can be predicted that might not be evident to the experimenter. This requires precise mathematical models. Because of the complexity of the living systems, theoretical biology employs several fields of mathematics, 5, and has contributed to the development of new techniques. History. Early history. Mathematics has been used in biology as early as the 13th century, when Fibonacci used the famous Fibonacci series to describe a growing population of rabbits. In the 18th century, Daniel Bernoulli applied mathematics to describe the effect of smallpox on the human population. Thomas Malthus' 1789 essay on the growth of the human population was based on the concept of exponential growth. Pierre-François Verhulst formulated the logistic growth model in 1836. Fritz Muller described the evolutionary benefits of what is now called Mullerian mimicry in 1879, in an account notable for being the first use of a mathematical argument in evolutionary ecology to show how powerful the effect of natural selection would be, unless one includes Malthus's discussion of the effects of population growth that influenced Charles Darwin. Malthus argued that growth would be exponential. He uses the word geometric, while resources, the environment's carrying capacity, could only grow arithmetically. 6. The term, theoretical biology, was first used as a monograph title by Johannes Reinke in 1901, and soon after by Jakob von Uexküll in 1920. One founding text is considered to be On Growth and Form, 1917, by Darcy Thompson. 7. And other early pioneers include Ronald Fisher, Hans Leo Abram, Vito Volterra, Nicholas Rashevsky and Conrad Hal Waddington. 8. Recent growth. Interest in the field has grown rapidly from the 1960s onwards. Some reasons for this include the rapid growth of data-rich information sets, due to the genomics revolution, which are difficult to understand without the use of analytical tools. 9. Recent development of mathematical tools such as chaos theory to help understand complex, nonlinear mechanisms in biology. An increase in computing power, which facilitates calculations and simulations not previously possible. An increasing interest in silico experimentation due to ethical considerations, risk, unreliability and other complications involved in human and non-human animal research. Areas of research. Several areas of specialized research in mathematical and theoretical biology 10, 11, 12, 13, 14, as well as external links to related projects in various universities are concisely presented in the following subsections, including also a large number of appropriate validating references from a list of several thousands of published authors contributing to this field. Many of the included examples are characterized by highly complex, nonlinear, and supercomplex mechanisms, as it is being increasingly recognized that the result of such interactions may only be understood through a combination of mathematical, logical, physical, chemical, molecular and computational models. Abstract relational biology. Abstract relational biology (ARB) is concerned with the study of general, relational models of complex biological systems, usually abstracting out specific morphological, or anatomical, structures. Some of the simplest models in ARB are the metabolic replication, or M.R., systems introduced by Robert Rosen in 1957-1958 as abstract, relational models of cellular and organismal organization. Other approaches include the notion of autopoiesis developed by Maturana and Varela, Kaufman's work Constraint Cycles, and more recently the notion of closure of constraints. 15. Algebraic biology. Algebraic biology, also known as symbolic systems biology, applies the algebraic methods of symbolic computation to the study of biological problems, especially in genomics, proteomics, analysis of molecular structures and study of genes. 16, 17, 18. Complex systems biology. An elaboration of systems biology to understand the more complex life processes was developed since 1970 in connection with molecular set theory, relational biology and algebraic biology. Computer models and automata theory. A monograph on this topic summarizes an extensive amount of published research in this area up to 1986, 2021, including subsections in the following areas: computer modeling in biology and medicine, arterial system models, neuron models, biochemical and oscillation networks, quantum automata, quantum computers in molecular biology and genetics, 22 cancer modeling, 23 neural nets, genetic networks, abstract categories in relational biology, 24 metabolic replication systems, category theory, 25 applications in biology and medicine. 26 automata theory, cellular automata, 27 tessellation models, 28, 29, and complete self-reproduction, chaotic systems in organisms, relational biology and organismic theories. 16, 30. Modeling cell and molecular biology. This area has received a boost due to the growing importance of molecular biology. 13 mechanics of biological tissues, 31, 32. Theoretical enzymology and enzyme kinetics. Cancer modeling and simulation, 33, 34. Modeling the movement of interacting cell populations, 35 mathematical modeling of scar tissue formation, 36 mathematical modeling of intracellular dynamics, 37, 38 mathematical modeling of the cell cycle, 39 mathematical modeling of apoptosis, 40 modeling physiological systems, modeling of arterial disease, 41 multi-scale modeling of the heart, 42 modeling electrical properties of muscle interactions, as in bidomain and monodomain models. Computational neuroscience. Computational neuroscience, also known as theoretical neuroscience or mathematical neuroscience, is the theoretical study of the nervous system. 43, 44. Evolutionary biology. Ecology and evolutionary biology have traditionally been the dominant fields of mathematical biology. Evolutionary biology has been the subject of extensive mathematical theorizing. The traditional approach in this area, which includes complications from genetics, is population genetics. Most population geneticists consider the appearance of new alleles by mutation, the appearance of new genotypes by recombination, and changes in the frequencies of existing alleles and genotypes at a small number of gene loci. When infinitesimal effects at a large number of gene loci are considered, together with the assumption of linkage equilibrium or quasi-linkage equilibrium, one derives quantitative genetics. Ronald Fisher made fundamental advances in statistics, such as analysis of variance, via his work on quantitative genetics. Another important branch in genetics that led to the extensive development of coalescent theory is phylogenetics. Phylogenetics is an area that deals with the reconstruction and analysis of phylogenetic, evolutionary, trees and networks based on inherited characteristics. 45 Traditional population genetic models deal with alleles and genotypes, and are frequently stochastic. Many population genetics models assume that population sizes are constant. Variable population sizes, often in the absence of genetic variation, are treated by the field of population dynamics. Work in this area dates back to the 19th century, and even as far as 1798 when Thomas Malthus formulated the first principle of population dynamics, which later became known as the Malthusian growth model. The Lotka-Volterra predator-prey equations are another famous example. Population dynamics overlap with another active area of research in mathematical biology, mathematical epidemiology, the study of infectious disease affecting populations. Work in this area dates back to the 19th century, and even as far as 1798 when Thomas Malthus formulated the first principle of population dynamics, which later became known as the Malthusian growth model. The Lotka-Volterra predator-prey equations are another famous example. Population dynamics overlap with another active area of research in mathematical biology, mathematical epidemiology, the study of infectious disease affecting populations. Various models of the spread of infections have been proposed and analyzed, and provide important results that may be applied to health policy decisions. In evolution, In evolutionary game theory, developed first by John Maynard Smith and George R. Price, selection acts directly on inherited phenotypes, without genetic complications. This approach has been mathematically refined to produce the field of adaptive dynamics. Mathematical biophysics. The earlier stages of mathematical biology were dominated by mathematical biophysics, described as the application of mathematics in biophysics, often involving specific physical, mathematical models of biosystems and their components or compartments. The following is a list of mathematical descriptions and their assumptions. Deterministic processes, dynamical systems. A fixed mapping between an initial state and a final state. Starting from an initial condition and moving forward in time, a deterministic process always generates the same trajectory, and no two trajectories cross in state space. Difference equations, maps, discrete time, continuous state space. Ordinary differential equations, continuous time, continuous state space, no spatial derivatives. See also, numerical ordinary differential equations. Partial differential equations, continuous time, continuous state space, spatial derivatives. See also, numerical partial differential equations. Logical deterministic cellular automata, discrete time, discrete state space. See also, cellular automaton. Stochastic processes, random dynamical systems. A random mapping between an initial state and a final state, making the state of the system a random variable with a corresponding probability distribution. Non-Markovian processes, generalized master equation, continuous time with memory of past events, discrete state space, waiting times of events or transitions between states discretely occur. Jump Markov process, master equation, continuous time with no memory of past events, discrete state space, waiting times between events discretely occur and are exponentially distributed. See also, Monte Carlo method for numerical simulation methods, specifically dynamic Monte Carlo method and Gillespie algorithm. Continuous Markov process, stochastic differential equations or a Fokker-Planck equation, continuous time, continuous state space, events occur continuously according to a random Wiener process. Spatial modeling. One classic work in this area is Alan Turing's paper on morphogenesis entitled The Chemical Basis of Morphogenesis, published in 1952 in the Philosophical Transactions of the Royal Society. Traveling Waves in a Wound Healing Assay, 46. Swarming Behavior, 47. A Mechanochemical Theory of Morphogenesis, 48. Biological Pattern Formation, 49. Spatial Distribution Modeling using Plot Samples, 50. Turing Patterns, 51. Mathematical Methods. A model of a biological system is converted into a system of equations, although the word model is often used synonymously with a system of corresponding equations. The solution of the equations, by either analytical or numerical means, describes how the biological system behaves either over time or at equilibrium. There are many different types of equations and the type of behavior that can occur is dependent on both the model and the equations used. The model often makes assumptions about the system. The equations may also make assumptions about the nature of what may occur. Molecular set theory. Molecular set theory, MST, is a mathematical formulation of the wide-sense chemical kinetics of biomolecular reactions in terms of sets of molecules and their chemical transformations represented by set theoretical mappings between molecular sets. It was introduced by Anthony Bartholomé, and its applications were developed in mathematical biology and especially in mathematical medicine. 52. In a more general sense, MST is the theory of molecular categories defined as categories of molecular sets and their chemical transformations represented as set theoretical mappings of molecular sets. The theory has also contributed to biostatistics and the formulation of clinical biochemistry problems in mathematical formulations of pathological, biochemical changes of interest to physiology, clinical biochemistry and medicine. 52. Organizational biology. Theoretical approaches to biological organization aim to understand the interdependence between the parts of organisms. They emphasize the circularities that these interdependences lead to. Theoretical biologists developed several concepts to formalize this idea. For example, abstract relational biology (ARB 53) is concerned with the study of general, relational models of complex biological systems, usually abstracting out specific morphological, or anatomical, structures. Some of the simplest models in ARB are the metabolic replication, or MR. Systems introduced by Robert Rosen in 1957-1958 as abstract, relational models of cellular and organismal organization. 54. Model example, the cell cycle. The eukaryotic cell cycle is very complex and has been the subject of intense study, since its misregulation leads to cancers. It is possibly a good example of a mathematical model as it deals with simple calculus but gives valid results. Two research groups 55-56 have produced several models of the cell cycle simulating several organisms. They have recently produced a generic eukaryotic cell cycle model that can represent a particular eukaryote depending on the values of the parameters, demonstrating that the idiosyncrasies of the individual cell cycles are due to different protein concentrations and affinities, while the underlying mechanisms are conserved. Chikashnagy et al., 2006. Evolutionary biology. Ecology and evolutionary biology have traditionally been the dominant fields of mathematical biology. Evolutionary biology has been the subject of extensive mathematical theorizing. The traditional approach in this area, which includes complications from genetics, is population genetics. Most population geneticists consider the appearance of new alleles by mutation, the appearance of new genotypes by recombination, and changes in the frequencies of existing alleles and genotypes at a small number of gene loci. When infinitesimal effects at a large number of gene loci are considered, together with the assumption of linkage equilibrium or quasi-linkage equilibrium, one derives quantitative genetics. Ronald Fisher made fundamental advances in statistics, such as analysis of variance, via his work on quantitative genetics. Another important branch of population genetics that led to the extensive development of coalescent theory is phylogenetics. Phylogenetics is an area that deals with the reconstruction and analysis of phylogenetic, evolutionary, trees and networks based on inherited characteristics. 45 Traditional population genetic models deal with alleles and genotypes, and are frequently stochastic. Many population genetics models assume that population sizes are constant. Variable population sizes, often in the absence of genetic variation, are treated by the field of population dynamics. Work in this area dates back to the 19th century, and even as far as 1798 when Thomas Malthus formulated the first principle of population dynamics, which later became known as the Malthusian growth model. The Lotka-Volterra predator-prey equations are another famous example. Population dynamics overlap with another active area of research in mathematical biology, mathematical epidemiology, the study of infectious disease affecting populations. Various models of the spread of infections have been proposed and analyzed, and provide important results that may be applied to health policy decisions. In evolutionary game theory, developed first by John Maynard Smith and George R. Price, selection acts directly on inherited phenotypes, without genetic complications. This approach has been mathematically refined to produce the field of adaptive dynamics. Mathematical biophysics. The earlier stages of mathematical biology were dominated by mathematical biophysics, described as the application of mathematics in biophysics, often involving specific physical, mathematical models of biosystems and their components or compartments. The following is a list of mathematical descriptions and their assumptions. Deterministic processes, dynamical systems. A fixed mapping between an initial state and a final state. Starting from an initial condition and moving forward in time, a deterministic process always generates the same trajectory, and no two trajectories cross in state space. Difference equations, maps, discrete time, continuous state space. Ordinary differential equations, continuous time, continuous state space, no spatial derivatives. See also, numerical ordinary differential equations. Partial differential equations, continuous time, continuous state space, spatial derivatives. See also, numerical partial differential equations. Logical deterministic cellular automata, discrete time, discrete state space. See also, cellular automaton. Stochastic processes, random dynamical systems. A random mapping between an initial state and a final state, making the state of the system a random variable with a corresponding probability distribution. Non-Markovian processes, generalized master equation, continuous time with memory of past events, discrete state space, waiting times of events or transitions between states discretely occur. Jump Markov process, master equation, continuous time with no memory of past events, discrete state space, waiting times between events discretely occur and are exponentially distributed. See also, Monte Carlo method for numerical simulation methods, specifically dynamic Monte Carlo method and Gillespie algorithm. Continuous Markov process, stochastic differential equations or a Fokker-Planck equation, continuous time, continuous state space, events occur continuously according to a random Wiener process. Spatial modeling. One classic work in this area is Alan Turing's paper on morphogenesis entitled The Chemical Basis of Morphogenesis, published in 1952 in the Philosophical Transactions of the One classic work in this area is Alan Turing's paper on morphogenesis entitled The Chemical Basis of Morphogenesis, published in 1952 in the Philosophical Transactions of the Royal Society. Traveling Waves in a Wound Healing Assay, 46. Swarming Behavior, 47. A Mechanochemical Theory of Morphogenesis, 48. Biological Pattern Formation, 49. Spatial Distribution Modeling Using Plot Samples, 50. Turing Patterns, 51. Mathematical Methods. A model of a biological system is converted into a system of equations, although the word model is often used synonymously with a system of corresponding equations. The solution of the equations, by either analytical or numerical means, describes how the biological system behaves either over time or at equilibrium. There are many different types of equations and the type of behavior that can occur is dependent on both the model and the equations used. The model often makes assumptions about the system. The equations may also make assumptions about the nature of what may occur. Molecular set theory. Molecular set theory, MST, is a mathematical formulation of the wide-sense chemical kinetics of biomolecular reactions in terms of sets of molecules and their chemical transformations represented by set theoretical mappings between molecular sets. It was introduced by Anthony Bartholomé, and its applications were developed in mathematical biology and especially in mathematical medicine. 52. In a more general sense, MST is the theory of molecular categories defined as categories of molecular sets and their chemical transformations represented as set theoretical mappings of molecular sets. The theory has also contributed to biostatistics and the formulation of clinical biochemistry problems in mathematical formulations of pathological, biochemical changes of interest to physiology, clinical biochemistry and medicine. 52. Organizational biology. Theoretical approaches to biological organization aim to understand the interdependence between the parts of organisms. They emphasize the circularities that these interdependences lead to. Theoretical biologists developed several concepts to formalize this idea. For example, abstract relational biology, ARB 53, is concerned with the study of general, relational models of complex biological systems, usually abstracting out specific morphological, or anatomical, structures. Some of the simplest models in ARB are the metabolic replication, or MR. Systems introduced by Robert Rosen in 1957-1958 as abstract, relational models of cellular and organismal organization. 54. Model example, the cell cycle. The eukaryotic cell cycle is very complex and has been the subject of intense study, since its misregulation leads to cancers. It is possibly a good example of a mathematical model as it deals with simple calculus but gives valid results. Two research groups, 55-56, have produced several models of the cell cycle simulating several organisms. They have recently produced a generic eukaryotic cell cycle model that can represent a particular eukaryote depending on the values of the parameters, demonstrating that the idiosyncrasies of the individual cell cycles are due to different protein concentrations and affinities, while the underlying mechanisms are conserved. Chikash-Nagy et al., 2006. By means of a system of ordinary differential equations, these models show the change in time, dynamical system, of the protein inside a single typical cell. This type of model is called a deterministic process, whereas a model describing a statistical distribution of protein concentrations in a population of cells is called a stochastic process. To obtain these equations, an iterative series of steps must be done. First, the several models and observations are combined to form a consensus diagram and the appropriate kinetic laws are chosen to write the differential equations, such as rate kinetics for stoichiometric reactions, Michaelis-Menten kinetics for enzyme-substrate reactions and Goldbeeder-Koechlin kinetics for ultrasensitive transcription factors. Afterwards, the parameters of the equations, rate constants, enzyme efficiency coefficients and Michaelis constants must be fitted to match observations. When they cannot be fitted, the kinetic equation is revised, and when that is not possible, the wiring diagram is modified. The parameters are fitted and validated using observations of both wild type and mutants, such as protein half-life and cell size. To fit the parameters, the differential equations must be studied. This can be done either by simulation or by analysis. In a simulation, given a starting vector list of the values of the variables, the progression of the system is calculated by solving the equations at each time frame in small increments. In analysis, the properties of the equations are used to investigate the behavior of the system depending on the values of the parameters and variables. A system of differential equations can be represented as a vector field, where each vector describes the change in concentration of two or more protein, determining where and how fast the trajectory simulation is heading. Vector fields can have several special points. A stable point, called a sink, that attracts in all directions, forcing the concentrations to be at a certain value; an unstable point, either a source or a saddle point, which repels, forcing the concentrations to change away from a certain value; and a limit cycle, a closed trajectory towards which several trajectories spiral towards, making the concentrations oscillate. A better representation, which handles the large number of variables and parameters, is a bifurcation diagram using bifurcation theory. The presence of these special steady-state points at certain values of a parameter, e.g. mass, is represented by a point, and once the parameter passes a certain value, a qualitative change occurs, called a bifurcation, in which the nature of the space changes, with profound consequences for the protein concentrations. The cell cycle has phases partially corresponding to G1 and G2, in which mass, via a stable point, controls cyclin levels, and phases S and M phases, in which the concentrations change independently. But once the phase has changed at a bifurcation event, cell cycle checkpoint, the system cannot go back to the previous levels, since at the current mass the vector field is profoundly different and the mass cannot be reversed back through the bifurcation event, making a checkpoint irreversible. In particular, the S and M checkpoints are regulated by means of special bifurcations called a Hopf bifurcation and an infinite period bifurcation. Biological applications of bifurcation theory: biophysics, biostatistics, entropy in life. See also mathematics. Linear function. In mathematics, the term linear function refers to two distinct but related notions. 1. In calculus and related areas, a linear function is a function whose graph is a straight line, that is, a polynomial function of degree 0 or 1. 2. For distinguishing such a linear function from the other concept, the term affine function is often used. 3. In linear algebra, mathematical analysis, 4. And functional analysis, a linear function is a linear map. 5. As a polynomial function. Graphs of two linear functions. In calculus, analytic geometry and related areas, a linear function is a polynomial of degree 1 or less, including the zero polynomial, the latter not being considered to have degree 0. When the function is of only one variable, it is of the form where a and b are constants, often real numbers. The graph of such a function of one variable is a non-vertical line. A is frequently referred to as the slope of the line, and b is the intercept. If a greater than 0, then the gradient is positive and the graph slopes upwards. If a less than 0, then the gradient is negative and the graph slopes downwards. For a function of any finite number of variables, the general formula is, and the graph is a hyperplane of dimension k. A constant function is also considered linear in this context, as it is a polynomial of degree 0 or as the zero polynomial. Its graph, when there is only one variable, is a horizontal line. In this context, a function that is also a linear map, the other meaning, may be referred to as a homogeneous linear function or a linear form. In the context of linear algebra, the polynomial functions of degree 0 or 1 are the scalar value defined maps. As a linear map, the integral of a function is a linear map from the vector space of integrable functions to the real numbers. In linear algebra, a linear function is a map F between two vector spaces such that here it denotes a constant belonging to some field k of scalars, for example, the real numbers, and x and y are elements of a vector space, which might be k itself. In other terms, a linear function preserves vector addition and scalar multiplication. Some authors use linear function only for linear maps that take values in the scalar field. 6. These are more commonly called linear forms. The linear functions of calculus qualify as linear maps when and only when f(0) 0 equals 0, or, equivalently, when the constant b equals 0 in the 1-degree polynomial above. Geometrically, the graph of the function must pass through the origin. Linear. In mathematics, the term linear function refers to two distinct but related notions. 1. In calculus and related areas, a linear function is a function whose graph is a straight line, that is, a polynomial function of degree 0 or 1. 2. For distinguishing such a linear function from the other concept, the term affine function is often used. 3. In linear algebra, mathematical analysis, 4. And functional analysis, a linear function is a linear map. 5. As a polynomial function. Graphs of two linear functions. In calculus, analytic geometry and related areas, a linear function is a polynomial of degree 1 or less, including the zero polynomial, the latter not being considered to have degree 0. When the function is of only one variable, it is of the form where a and b are constants, often real numbers. The graph of such a function of one variable is a non-vertical line. A is frequently referred to as the slope of the line, and b is the intercept. If a greater than 0, then the gradient is positive and the graph slopes upwards. If a less than 0, then the gradient is negative and the graph slopes downwards. For a function of any finite number of variables, the general formula is, and the graph is a hyperplane of dimension k. A constant function is also considered linear in this context, as it is a polynomial of degree 0 or as the zero polynomial. Its graph, when there is only one variable, is a horizontal line. In this context, a function that is also a linear map, the other meaning, may be referred to as a homogeneous linear function or a linear form. In the context of linear algebra, the polynomial functions of degree 0 or 1 are the scalar value defined maps. As a linear map, the integral of a function is a linear map from the vector space of integrable functions to the real numbers. In linear algebra, a linear function is a map f between two vector spaces such that here it denotes a constant belonging to some field k of scalars, for example, the real numbers and x and y are elements of a vector space, which might be k itself. In other terms, a linear function preserves vector addition and scalar multiplication. Some authors use linear function only for linear maps that take values in the scalar field. 6. These are more commonly called linear forms. The linear functions of calculus qualify as linear maps when and only when f(0, 0 equals 0, or, equivalently, when the constant b equals 0 in the 1-degree polynomial above. Geometrically, the graph of the function must pass through the origin. Quadratic equation. In mathematics, a quadratic equation, from Latin quadratus square, is an equation that can be rearranged in standard form as 1, where the variable x represents an unknown number, and a, b, and c represent known numbers, where a does not equal 0. If a equals 0 and b does not equal 0. Quadratic equation. In mathematics, a quadratic equation, from Latin quadratus square, is an equation that can be rearranged in standard form as 1, where the variable x represents an unknown number, and a, b, and c represent known numbers, where a does not equal 0. If a equals 0 and b does not equal 0, then the equation is linear, not quadratic. The numbers a, b, and c are the coefficients of the equation and may be distinguished by respectively calling them the quadratic coefficient, the linear coefficient, and the constant coefficient or free term. 2. The values of x that satisfy the equation are called solutions of the equation, and roots or zeros of the quadratic function on its left-hand side. A quadratic equation has at most two solutions. If there is only one solution, one says that it is a double root. If all the coefficients are real numbers, there are either two real solutions, or a single real double root, or two complex solutions that are complex conjugates of each other. A quadratic equation always has two roots. If complex roots are included and a double root is counted for 2. A quadratic equation can be factored into an equivalent equation. 3. Where r and s are the solutions for x. The quadratic formula expresses the solutions in terms of a, b, and circa. Completing the square is one of several ways for deriving the formula. Solutions to problems that can be expressed in terms of quadratic equations were known as early as 2000 BC. 4. 5. Because the quadratic equation involves only one unknown, it is called univariate. The quadratic equation contains only powers of x that are non-negative integers, and therefore it is a polynomial equation. In particular, it is a second-degree polynomial equation, since the greatest power is 2. Solving the quadratic equation. Figure 1. Plots of quadratic function y equals x2 plus bx plus c, varying each coefficient separately while the other coefficients are fixed, at values a equals 1, b equals 0, c equals 0. A quadratic equation whose coefficients are real numbers can have either 0, 1, or two distinct real-valued solutions, also called roots. When there is only one distinct root, it can be interpreted as two roots with the same value, called a double root. When there are no real roots, the coefficients can be considered as complex numbers with zero imaginary part, and the quadratic equation still has two complex-valued roots, complex conjugates of each other with a non-zero imaginary part. A quadratic equation whose coefficients are arbitrary complex numbers always has two complex-valued roots which may or may not be distinct. The solutions of a quadratic equation can be found by several alternative methods. Factoring by inspection. It may be possible to express a quadratic equation x2 plus bx plus c equals 0 as a product px plus q rx plus s equals 0. In some cases, it is possible, by simple inspection, to determine values of p, q, r, and s that make the two forms equivalent to one another. If the quadratic equation is written in the second form, then the zero-factor property states that the quadratic equation is satisfied if px plus q equals 0 or rx plus s equals 0. Solving these two linear equations provides the roots of the quadratic. For most students, factoring by inspection is the first method of solving quadratic equations to which they are exposed. 6. 202-207 If one is given a quadratic equation in the form x2 plus bx plus c equals 0, the sought factorization has the form x plus q x plus s, and one has to find two numbers q and s that add up to b and whose product is c. This is sometimes called Vieta's rule, 7, and is related to Vieta's formulas. As an example, x2 plus 5x plus 6 factors is x plus 3, x plus 2. The more general case where it does not equal 1 can require a considerable effort in trial and error guess and check, assuming that it can be factored at all by inspection. Except for special cases such as where b equals 0 or c equals 0, factoring by inspection only works for quadratic equations that have rational roots. This means that the great majority of quadratic equations that arise in practical applications cannot be solved by factoring by inspection. 6. 207. Completing the square. Figure 2. For the quadratic function y equals x2 minus x minus 2, the points where the graph crosses the x-axis, x equals minus 1 and x equals 2, are the solutions of the quadratic equation x2 minus x minus 2 equals 0. The process of completing the square makes use of the algebraic identity. which represents a well-defined algorithm that can be used to solve any quadratic equation. 207 Starting with a quadratic equation in standard form, x2 plus bx plus c equals 0. 1. Divide each side by a, the coefficient of the squared term. 2. Subtract the constant term c, a from both sides. 3. Add the square of 1/2 of b, a, the coefficient of x, to both sides. This completes the square, converting the left side into a perfect square. 4. Write the left side as a square and simplify the right side if necessary. 5. Produce two linear equations by equating the square root of the left side with the positive and negative square roots of the right side. 6. Solve each of the two linear equations. We illustrate use of this algorithm by solving 2x2 plus 4x minus 4 equals 0. The plus minus symbol, plus or minus, indicates that both are solutions of the quadratic equation. 8. Quadratic formula and its derivation. Completing the square can be used to derive a general formula for solving quadratic equations, called the quadratic formula. 9th, the mathematical proof will now be briefly summarized. 10th, it can easily be seen, by polynomial expansion, that the following equation is equivalent to the quadratic equation. Taking the square root of both sides, and isolating x, gives. Some sources, particularly older ones, use alternative parametrizations of the quadratic equation such as x2 plus 2bx plus c equals 0 or x2 minus 2bx plus c equals 0. 11. Where b has a magnitude 1/2 of the more common one, possibly with opposite sign. These result in slightly different forms for the solution, but are otherwise equivalent. A number of alternative derivations can be found in the literature. These proofs are simpler than the standard completing the square method, represent interesting applications of other frequently used techniques in algebra, or offer insight into other areas of mathematics. A lesser-known quadratic formula, as used in Muller's method, provides the same roots via the equation. This can be deduced from a standard quadratic formula by Vieta's formulas, which assert that the product of the roots is CA. It also follows from dividing the quadratic equation by solving this for and then inverting. One property of this form is that it yields one valid root when a equals 0, while the other root contains division by 0, because when a equals 0, the quadratic equation becomes a linear equation, which has one root. By contrast, in this case, the more common formula has a division by 0 for one root and an indeterminate form 0/0 for the other root. On the other hand, when c equals 0, the more common formula yields two correct roots, whereas this form yields the 0 root and an indeterminate form 0/0. When neither a nor c is 0, the equality between the standard quadratic formula and Muller's method can be verified by cross-multiplication, and similarly for the other choice of signs. Reduced quadratic equation. It is sometimes convenient to reduce a quadratic equation so that its leading coefficient is 1. This is done by dividing both sides by a, which is always possible since a is non-zero. This produces the reduced quadratic equation, 12. Where p equals b, a and q equals c, a. This monic polynomial equation has the same solutions as the original. The quadratic formula for the solutions of the reduced quadratic equation, written in terms of its coefficients, is discriminant. Figure 3. Discriminant signs. In the quadratic formula, the expression underneath the square root sign is called the discriminant of the quadratic equation, and is often represented using an uppercase d or an uppercase Greek delta. 13. A quadratic equation with real coefficients can have either one or two distinct real roots, or two distinct complex roots. In this case, the discriminant determines the number and nature of the roots. There are three cases. If the discriminant is positive, then there are two distinct roots, both of which are real numbers. For quadratic equations with rational coefficients, if the discriminant is a square number, then the roots are rational. In other cases they may be quadratic irrationals. If the discriminant is 0, then there is exactly one real root, sometimes called a repeated or double root, or two equal roots. If the discriminant is negative, then there are no real roots. Rather, there are two distinct, non-real, complex roots, 14, which are complex conjugates of each other. In these expressions I is the imaginary unit. Thus the roots are distinct if and only if the discriminant is non-zero, and the roots are real if and only if the discriminant is non-negative. Geometric interpretation. Visualization of the complex roots of y equals x2 plus bx plus c. The parabola is rotated 180 degrees about its vertex, orange. Its x-intercepts are rotated 90 degrees around their midpoint, and the Cartesian plane is interpreted as the complex plane, green. 15. The function f x equals x2 plus bx plus c is a quadratic function. 16. The graph of any quadratic function has the same general shape, which is called a parabola. The location and size of the parabola, and how it opens, depend on the values of a, b, and circa. If a greater than 0, the parabola has a minimum point and opens upward. If a less than 0, the parabola has a maximum point and opens downward. The extreme point of the parabola, whether minimum or maximum, corresponds to its vertex. The x-coordinate of the vertex will be located at, and the y-coordinate of the vertex may be found by substituting this x value into the function. The y-intercept is located at the point 0, c. The solutions of the quadratic equation x2 plus bx plus c equals 0 correspond to the roots of the function f x equals x2 plus bx plus c, since they are the values of x for which f x equals 0. If a, b, and c are real numbers and the domain of f is the set of real numbers, then the roots of f are exactly the x-coordinates of the points where the graph touches the x-axis. If the discriminant is positive, the graph touches the x-axis at 2 points. If 0, the graph touches at 1 point, and if negative, the graph does not touch the x-axis. Quadratic factorization. The term is a factor of the polynomial. If and only if r is a root of the quadratic equation. It follows from the quadratic formula that in the special case v2 equals 4ac where the quadratic has only one distinct root, i.e. the discriminant is 0, the quadratic polynomial can be factored as. Graphical solution. Figure 4, graphing calculator. Computation of one of the two roots of the quadratic equation 2x2 plus 4x minus 4 equals 0. Although the display shows only 5 significant figures of accuracy, the retrieved value of xc is 0.732050807569, accurate to 12 significant figures. A quadratic function without real root, y equals x minus 5 2 plus 9. The 3 is the imaginary part of the x-intercept. The real part is the x-coordinate of the vertex. Thus the roots are 5 plus or minus 3i. The solutions of the quadratic equation may be deduced from the graph of the quadratic function, which is a parabola. If the parabola intersects the x-axis in 2 points, there are 2 real roots, which are the x-coordinates of these 2 points, also called x-intercept. If the parabola is tangent to the x-axis, there is a double root, which is the x-coordinate of the contact point between the graph and parabola. If the parabola does not intersect the x-axis, there are 2 complex conjugate roots. Although these roots cannot be visualized on the graph, their real and imaginary parts can be. 17. Let h and k be respectively the x-coordinate and the y-coordinate of the vertex of the parabola, that is the point with maximal or minimal y-coordinate. The quadratic function may be rewritten. Let d be the distance between the point of y-coordinate 2k on the axis of the parabola, and a point on the parabola with the same y-coordinate. See the figure. There are 2 such points, which give the same distance, because of the symmetry of the parabola. Then the real part of the roots is h, and their imaginary part are plus or minus d. That is, the roots are. Or in the case of the example of the figure. Avoiding loss of significance. Although the quadratic formula provides an exact solution, the result is not exact if real numbers are approximated during the computation, as usual in numerical analysis, where real numbers are approximated by floating point numbers called reals in many programming languages. In this context, the quadratic formula is not completely stable. This occurs when the roots have different order of magnitude, or, equivalently, when b2 and b2 minus 4ac are close in magnitude. In this case, the subtraction of 2 nearly equal numbers will cause loss of significance or catastrophic cancellation in the smaller root. To avoid this, the root that is smaller in magnitude, r, can be computed as where r is the root that is bigger in magnitude. This is equivalent to using the formula. Using the plus sign if and the minus sign if. A second form of cancellation can occur between the terms b2 and 4ac of the discriminant, that is when the two roots are very close. This can lead to loss of up to half of correct significant figures in the roots. 11, 18. Examples and applications. The trajectory of the cliff jumper is parabolic because horizontal displacement is a linear function of time, while vertical displacement is a quadratic function of time. As a result, the path follows quadratic equation, wherein are horizontal and vertical components of the original velocity, a is gravitational acceleration and h is original height. The value should be considered negative here, as its direction downwards is opposite to the height measurement upwards. The golden ratio is found as the positive solution of the quadratic equation. The equations of the circle and the other conic sections, ellipses, parabolas, and hyperbolas, are quadratic equations in 2 variables. Given the cosine or sine of an angle, finding the cosine or sine of the angle that is half as large involves solving a quadratic equation. The process of simplifying expressions involving the square root of an expression involving the square root of another expression involves finding the 2 solutions of a quadratic equation. Descartes' theorem states that for every 4 kissing, mutually tangent circles, their radii satisfy a particular quadratic equation. The equation given by Fuss' theorem, giving the relation among the radius of a bicentric quadrilateral's inscribed circle, the radius of its circumscribed circle, and the distance between the centers of those circles, can be expressed as a quadratic equation for which the distance between the 2 circles' centers in terms of their radii is one of the solutions. The other solution of the same equation in terms of the relevant radii gives the distance between the circumscribed circle's center and the center of the excircle of an x-tangential quadrilateral. Critical points of a cubic function and inflection points of a quartic function are found by solving a quadratic equation. In physics, for motion with constant acceleration, the displacement or position of a moving body can be expressed as a quadratic function of time given the initial position and initial velocity. In chemistry, the pH of a solution of weak acid can be calculated from the negative base 10 logarithm of the positive root of a quadratic equation in terms of the acidity constant and the analytical concentration of the acid. History. Babylonian mathematicians, as early as 2000 BC, displayed on old Babylonian clay tablets, could solve problems relating the areas and sides of rectangles. There is evidence dating this algorithm as far back as the 3rd dynasty of Ur. 19. In modern notation, the problems typically involve solving a pair of simultaneous equations of the form, which is equivalent to the statement that x and y are the roots of the equation. 2086. The steps given by Babylonian scribes for solving the above rectangle problem, in terms of x and y, were as follows. 1. Compute half of p. 2. Square the result. 3. Subtract q. 4. Find the positive square root using a table of squares. 5. Add together the results of steps 1 and 4 to give x. In modern notation this means calculating, which is equivalent to the modern-day quadratic formula for the larger real root, if any, with a equals 1. B equals minus P, and C equals Q. Geometric methods were used to solve quadratic equations in Babylonia, Egypt, Greece, China, and India. The Egyptian Berlin Papyrus, dating back to the Middle Kingdom 2050 BC to 1650 BC, contains the solution to a two-term quadratic equation.